We will be going over the sonic hedgehog pathway and its role in teeth development in Mammalia. So, how is it possible for this little guy, a hedgehog, to help form teeth in humans? Hedgehog signaling was first discovered in the 1970s in fruit flies. Scientists found that specific changes or mutations to a gene caused a developing fruit fly to curl up and develop bristly hair-like features resembling a hedgehog. A similar signaling pathway was found in humans, and so the name was carried over. The hedgehog pathway is activated during the early stage of life and is essential for the normal development of a growing embryo. We will be going over some of this terminology throughout the video. All of these play a part in the sonic hedgehog pathway. The hedgehog signaling molecule is a paracrine signal that is released by Doan cells. It functions on a membrane to the cell adjacent to it. It deals with the function of a gene that specifies tissue patterns in certain organisms. It also helps directly with developing different embryonic cells, including the epithelial cells for teeth morphogenesis and differentiation. There is this protein called patched, and what patched normal function is to inhibit a protein called smoothened. When smoothened is inhibited by patched, smoothened cannot exert its function. If smoothened becomes activated, its function involves inhibiting a protein called GLI3R and activates GLI2, GLI3A. Activating these two proteins are necessary to activate GLI1, which will bring up the sonic hedgehog target genes. If smoothened is inhibited, then there will be no activation of GLI2, GLI3A, and GLI1, and so activation of sonic hedgehog target genes will not happen and will stay off. In order to inhibit patched and activate smoothened, another cell will secrete an immature protein called pre-hedgehog. Pre-hedgehog has to be acetylated. It has to have palmitoyl-CoA fatty acid tail and is catalyzed by the hedgehog acetyltransferase, or HHAT. The HHAT uses the palmitoyl-CoA to transfer it to the pre-hedgehog, making a monomer of hedgehog, or HH1. Two hedgehog monomers have to dimerize into a hedgehog dimer called HH2. Once dimerization occurs, the other cell will secrete hedgehog2 into the ECF as a paracrine signal with the help of the protein dispatched, or DISP, and will bind to patched. Hedgehog2 acts as an inhibitor once it binds to patched. When you inhibit the inhibition of smoothened will become activated in the cilia, and then GLI2, GLI3A are directly activated, which will phosphorylate GLI1. Smoothened also inhibits GLI3R. The GLI3R function is to inhibit GLI2, GLI3A, but smoothened acts as an inhibitor to inhibit GLI3R's function. GLI1 will act as a transcription factor, and so GLI1 will go to the nucleus and then bind to the promoter region where the hedgehog target genes are located, and then it will upregulate those genes. Transcription of those genes and then translation will occur. All of this is super easy to understand through a simple analogy. Sonic is the name of a fast food restaurant chain and can be compared to a nucleus, where the sonic hedgehog target genes are expressed if activated. The smoothened protein can be compared to the typical college student wanting to go out to buy food from Sonic. Patched can be compared to a disapproving parent who inhibits smoothened from going to Sonic. GLI3R is equivalent to a big brother who also disapproves of you going out to get Sonic. Both patched and GLI3R express inhibition that prevents the function of GLI2, GLI3A, which is like your car to get to Sonic. GLI1 will not be able to be expressed, and the sonic hedgehog target genes in the nucleus will not be obtained, or in this case, expressed. To inhibit patched, aka the parent inhibiting you to go to Sonic, the pre-hedgehog is comparable to a report card showing straight A's. When the hedgehog molecule binds to patched, it's just as if the parent saw you had good grades and will stop inhibiting smoothened from going to Sonic. The report card, or hedgehog, can be shown to the big brother as well, or Glee3R, who will also approve of you going to Sonic and will stop inhibiting Glee2, Glee3A, which is the car. Glee1 is able to be expressed and the Sonic restaurant is there for you to get. So now you're wondering, how does any of this relate to how teeth form? As mentioned earlier, the Sonic Hedgehog pathway also relates to the development of teeth. Epithelia contributes to embryonic development as well as the development and function of the body. Toothed Sonic Hedgehog develops not only teeth, but also things like nails as well. With the tooth germ being made up of mesenchymal tissues and epithelial tissues, Sonic Hedgehog plays a role in the epithelial signals and epithelial stromal signals found locally during crown formation. The mesenchyme initiates the Sonic Hedgehog pathway to create the epithelium. Differentiation occurs in the enamel organ and makes way for crown formation. Then, tooth morphogenesis in the enamel organ takes place during the calcification stage. Sonic Hedgehog expression is found in the Hertwig's epithelial root sheath, or HERS, and facilitates the last formation of the root. Further research is showing how involved Sonic Hedgehog signaling is in odontogenesis, especially in mammals like mice. When the formation of the crown occurs, the outer and inner epithelium of the enamel fuse, forming the HERS. The HERS is set between the dental papilla and the dental follicle and controls the formation of the root. Because of this mutation, the tissue disappears and the tooth root no longer elongates. In the first diagram, the different pictures show the stages of tooth development with transcription of Sonic Hedgehog. The second diagram shows the formation of different molars with different Sonic Hedgehog mutated embryos. There are numerous functions of the Sonic Hedgehog pathway. With greater understanding of tooth formation and new research on signaling molecules in the pathway, such as Glee1, as well as Sonic Hedgehog mutations that can occur, new potential dental therapies and processes for teeth restoration can be made. Thank you.